what this quote was saying is that this study that we just read is giving evidence that by knocking out MAD2 and BUB-R1, which are spindle checkpoint proteins, this creates a dysfunctional spindle checkpoint process within the MCF7 breast cancer cells. And as a result of the dysfunctional spindle checkpoint in these MCF7 breast cancer cells, they are resistant to the drug called paclitaxel. And paclitaxel is a drug that is used to treat cancer. So in this case, when these cancer cells have these two checkpoint proteins knocked out, they become resistant to paclitaxel. And the second question is, how can we demonstrate that we could knock down or downregulate MAD2 and BUB-R1 in a group of cells? The first technique that we would use in order to answer that question is siRNA. And we would have two groups of cells. So we'd have a control group, and then we would have a group of cells that would be treated with, let's just use BUB-R1 as the first example. We could do, it would be the same situation for MAD2, except MAD2 would be in this group of cells and there would still be a control. But we would have BUB1 siRNA being transfected into these cells. And this group of cells will just have a control vector. The reason why we would do this is because siRNA is a piece of RNA that's able to bind specific mRNA. So in this case, the siRNA that we made will bind to the BUB1 or BUB-R1 mRNA in these cells, and therefore decreasing the amount of mRNA within these cells. And as a result of the decrease in mRNA, there will be less BUB-R1 protein made in these cells. And that's the basic theory to siRNA. So after both of these cells are transfected, we can wait 24 hours to let this transfection occur, or let the siRNA do its thing. And in order to check the results, we could do a Western blot. So this would be the second technique that would be needed in order to demonstrate that we have downregulated MAD2 and or BUB-R1. And what we could really do in this case scenario is just have the control group of cells and the BUB-R1 siRNA cells. And as I said earlier, this could be MAD2 siRNA cells. It would be the same technique, but we're just going to focus on the BUB-R1. And we would check for the amount of BUB-R1 protein. This is BUB-R1. So you would expect to have a darker band in the Western blot for the control cells in comparison to the BUB-R1 siRNA cells. And that's because the primary and secondary antibodies that are made in order to bind to the BUB-R1 in these cells will detect more BUB-R1 or more BUB-R1 in the control cells, because there is no siRNA to take out the BUB-R1 mRNA. So if there's more BUB-R1 mRNA in the control cells, you would expect to see a darker band. And that would be the same situation if this just said MAD2. So you get the idea. What would you expect for question 3, the cell cycle to look like for, let's just say, the cells that have been treated with BUB-R1 siRNA? So as we said earlier, BUB-R1 is involved in a spindle checkpoint. It has a function in the spindle checkpoint process. And that basically is specifically telling us that BUB-R1 is most likely involved in mitosis, because mitosis has spindles and spindle fibers. And specifically in this case, we know just from past knowledge in class, BUB-R1 is specifically involved in the cell cycle, moving cells from metaphase to anaphase. So this is metaphase with the M, and the anaphase is an A. So I guess we'll first draw what a normal cell cycle will look like, and then we can draw the one with siRNA after. So in a normal cell, what will happen is the chromosomes will just line up as they should in a perfect orientation along the metaphase plate in the middle. And as they move into anaphase, these spindle fibers are going to pull these chromosomes apart, getting the cell ready to divide and actually replicate, which is coming soon in telophase and cytokinesis. And these spindle fibers will soon pull chromosomes away from each other, away from the metaphase plate. And ideally, there will be, one copy of chromosome 1, one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 2, all the way down to 23. So each spindle will have one copy of each chromosome pulled away. Now, what would happen if, let's say, BubR1 was knocked out? This process of pulling the chromosomes apart could potentially happen prematurely or happen incorrectly. And what would that mean? Well, as I said earlier, each cell should get one chromosome. And maybe in this case, when BubR1 isn't there, maybe the left side of right here, maybe there's 2 copies of chromosome 1 and this cell doesn't get any chromosome 1, or this group of spindle fibers doesn't have chromosome 1. So when the cell goes to divide, maybe this cell has 24 chromosomes, this one has 22, and as a result, that's aneuploidy right there. And that is directly correlated to just not having BubR1 regulating this movement from metaphase to anaphase. So any premature movement can just cause dysregulation in this process that I just described right here.